Alrighty. Hi, everyone, and welcome to the next session in the WRRC Spring Seminar Series. And today we have Dr. Barbara Bekins. She'll be talking about natural source zone depletion of petroleum hydrocarbons at the water table. Just to introduce our speaker, Dr. Barbara Bekins is a research hydrologist with the U.S. Geological Survey, water mission area located in Menlo Park, California. She has studied the fate and transport of crude oil contaminants in groundwater for over 25 years. Barbara serves as research coordinator for the USGS crude oil spill study site near Bemidji, Minnesota. She has been elected a fellow of the Geological Society of America and the American Geophysical Union, and is a member of the National Academy of Engineering. So Barbara, please go ahead.
Great, thanks so much for inviting me to this seminar series. I spoke in this series in 2018 in June, and the material I'm going to be presenting today is newer since that talk. So this talk is pretty much different from that one, but I will give you an introduction to the site, which, as Carrie said, I've been studying for over 25 years, and the USGS has been studying it for over almost 40 years. So I would like, I hope that this talk will provide some reassurance to you there that some of what will be happening to the fuel are things that we understand, and that the knowledge that I give to you today will help you understand some tools that we can use, and some ways to think about the fuel, and also some things that we don't know but are within reach, and our technology is giving us lots of opportunities to get a better understanding of these problems. So the talk will start with some background material about the spill that occurred in Minnesota and the response to it, and what the USGS study motivation is. And then I will move on to the crude oil source, and I'll try to connect it to the source that is the spilled fuel at Red Hill and how it changes with time. And then the third part will be about the contaminant plume that's in the groundwater, that and I'll cover the nature of the organic carbon that's in the plume, and also some biological test results we've been doing. So this is an aerial photo of the Bemidji site. It's located in northern Minnesota. At the time of the spill, there was a pipeline break. There's a pipeline running through the site, carries oil from Alberta, Canada, over to the Great Lakes. The spill, the pipeline was under pressure when it burst, and so the oil sprayed out of this over this area called the spray zone, and then it kind of ran down along the topography to this wetland here. And then they, in the emergency response, they trenched in order to recover some of the oil. So here's a scale bar to give you a sign of how big this is. This is a full-size tanker trailer here. So throughout the talk, I've got a few slides comparing Red Hill to Bemidji. So Red Hill is, of course, a layered basalt aquifer. Bemidji is a glacial outwash. So here's a cross-section, kind of big sandbox. This distance from the source to the water table at Red Hill is 80 to 100 feet. In Bemidji, it's about 20 feet. The recharge is quite high, at least in the mountains at Red Hill, compared to Bemidji. The groundwater velocity is 1 to 10 feet per day. It's quite a bit slower at Bemidji, just 0.2 feet per day. And the way to say you're welcome in Minnesotan is you betcha. And here it's Aoli Palikia. So here is what it looked like when they did the emergency response. There are the pipelines here, and the pipeline break was here, and this is the trenching that I showed you in the previous picture. So the emergency response included pumping of the oil, excavation and land farming, and burning or evaporation of oil-coated vegetation. And here we see some of the pumping of the oil. Interestingly, 1979, this guy's personal protective equipment was basically a do-rag. You know, it's kind of a different world than compared to today. The oil migrated into the subsurface quite rapidly. So of the 75% that didn't get cleaned up, about 25% soaked into these sandy sediments. And you can see it here seeping out the side. So here's an overview map of the site today and the important features that I'll be covering. So here was where the pipeline break was. Today, there are 6 oil pipelines running through the site. This is the area that was sprayed with oil. The groundwater flow is in this direction to the northeast. And the oil, as it flowed along the surface, these are the surface flow lines, infiltrated the aquifer in 3 locations. And the most studied location is this area. It's called the North Oil Pool. And wells that, the monitoring wells are shown here in green. There are hundreds of wells at the site to provide a research picture. The flow velocity is 5 to 50 centimeters per day. Now, the important thing about this site, and it was very successful in the early 1990s in demonstrating that benzene biodegrades naturally. There was no need to add anything to accelerate the biodegradation. And so the 10 microgram per liter contour of benzene in 1996 was here. And in 2010, this well still measured 3.3 micrograms per liter of benzene. So this aquifer is a very efficient bioreactor for benzene. And that is a success story that has been used to train us and create protocols for monitoring biodegradation of petroleum hydrocarbons in aquifers. So I'm not going to talk much about the benzene. I'm just going to go on to the newer work that we've been doing. The motivation for the study is that there are over 500,000 leaking underground storage tank releases nationwide. And those are managed with risk-based corrective action. So if there's no water at risk and the plume is stable or shrinking, then the site can be approved for no further action. And many sites are approved even though they still have contaminant remaining in the ground. So there are lots of sites around the country where fuel is still in the ground and the groundwater is monitored. And so this is public policy today, and it is working well for fuel sites across the country. But we have continued to study the Bemidji site for several reasons, and we've been funded by the USGS Toxic Substance Hydrology Program, and we're studying the fate, transport, and natural attenuation of crude oil. And we are tasked with providing the results to industry consultants and regulatory, educational, and research institutions like your own. And the reason why we've been continued to fund this problem, even though benzene, we have this success story about benzene, is that the oil is still in the ground, and these processes there are many, many processes that affect how the oil biodegrades and changes over time, including biodegradation, dissolution into the groundwater, and then there's a dissolved contaminant plume and transport of that contaminant plume. And then there are vapors that come also up towards the surface. So because the time frame is very long for natural biodegradation, and because there are so many spills where the source has been left in place for various reasons, we have successfully argued to management that this study should be ongoing. So just a little tutorial here about biodegradation. Reduced carbon, which includes spilled fuel and oil, can be oxidized by bacteria in redox reactions to obtain energy and create oxidized carbon. In some cases, that's carbon dioxide. In other cases, it is a more oxidized form than the parent carbon, and there's still energy available. So the most energetic reactions are coupled to oxygen, as is our redox reaction of burning food. In the absence of oxygen, and that occurs quite rapidly in the subsurface because oxygen is not very soluble in water, there are a series of electron acceptors utilized by bacteria, and these are grouped together as anaerobic processes. At the Bemidji site, the most important is iron, and then carbon dioxide in methanogenesis. But at Red Hill, the story is a little different. So here's some chemistry from Red Hill monitoring well 02 of the important electron acceptors. The blue is the data from the well that has typically shows some petroleum hydrocarbons in it. And the red is from a clean background well unaffected by the plume. So you can see dissolved oxygen is up here close to solubility, but at monitoring well 2, it's depleted, and that's indicating that there is biodegradation going on. Similarly, nitrate is depleted. Iron behaves the opposite. Iron is actually produced as bacteria consume iron oxide on the rocks, and then they put iron 2 into the aquifer. So you expect to see iron increased in the middle of the plume. Sulfate is very high because you're close to the ocean and the sulfate salts of the ocean, and it's depleted here. Methane is produced. So we know if methane is produced, then in some parts of the spill, all of these other electron acceptors are quite low in concentration because methane is inhibited by nitrate and oxygen and competes with sulfate. This is total organic carbon. In the clean aquifer, it's below 1 milligram per liter. And in the aquifer where that's impacted, it's higher. And I'll be talking a lot about total organic carbon in this talk. So now that you've had a tutorial on biodegradation, I'll just give you an overview of the Bemidji site and the many decades of results from that site. So the oil is here, present from land surface down to the water table. Within the oil itself, the most degradable compounds are the normal alkanes, and they degrade under methanogenic conditions to CO2 and methane. The methane outgasses and moves towards the surface, and it is biodegraded partway to the surface into CO2 and water. So there's no methane reaching the surface, but there's a lot of CO2 reaching the surface. And in fact, if you look at how much is biodegrading, we can show from that methane, that CO2 efflux, that 80%, 86% of the biodegraded carbon is leaving as CO2 at the land surface. So only 14% of the carbon actually enters the groundwater contaminant plume. And that's reflected here as the BTEX, so the the total petroleum hydrocarbon in the gasoline range. And then there's this dissolved organic carbon that isn't, and we call it non-volatile in order to distinguish it from BTEX, which is volatile. And that degrades close in under methanogenic conditions. And then out here at the advancing edge of the plume, there are iron oxides on the aquifer that the bacteria use in this redox reaction to oxidize the reduced carbon in the plume. So here's another comparison of Red Hill to Bemidji. So the spilled hydrocarbons at Bemidji were a crude oil, a light, low sulfur crude oil from Alberta. In 1979, an estimated 450,000 gallons were spilled. Within the North Pool, which is what my talk will cover, there are about 37,000 gallons. Here's a chromatogram of that, and this is retention time and response. The longer carbons are retained longer. So these are 25 carbon N alkanes, and this is 15. And so I've aligned these. This is a chromatogram of JP5, where the estimated volume is 19,000 gallons released in November. So this is retention time again. Actually, this is a carbon number here. So these are 10 carbons, 11, up to 15 carbons. So we don't have compounds that have up to 30 carbons like we have in crude oil in Bemidji. So that's one big difference in the two sites. So the first part of the talk is going to be on how the source changes over time. So in 2010, oil was collected from 13 wells that had oil floating at the water table. And then they were also sampled in a series multiple times. The oil was analyzed for volatile constituents. So the samples were collected from these wells shown in red. And then this is a cross section in the lower figure, and the well screens are, the position of the well screens are also shown in red. So the contour plot is oil saturation. So what that means is how much of the pore is filled with oil. So in this case, 50% of the pore is filled with oil, and then it drops towards the edge to less than 10%. So the wells intersect areas with varying degrees of oil saturation, and that is going to affect how things change. So in the next figure, I'm going to show you two contrasting wells with different saturations. One whose saturation is close to 0.5 and another whose saturation is closer to 0.3. So here are three compounds, and on the horizontal axis is time since the spill, and the vertical axis is fraction of that compound that's still remaining in the oil. So these are analyses of the oil. So we start out at the beginning with 100% of the benzene that's still in the oil. And then over time, it's dropped faster in this well shown in blue, where the surrounding pore space saturation is 37%, compared to this well shown in peach, where the surrounding oil saturation is almost 50%. And that's because the water contacts it more readily in areas where the oil saturation is lower. So in this case, even though it's contaminating the water is also serving to move the benzene into the bioreactor of the aquifer. So M-xylene, up here is the effective solubility. So this is the how many moles of benzene are dissolved in oil in equilibrium with water per mole of water. So this is the effective solubility. M-xylene has an order of magnitude lower solubility, and that's reflected in the fact that a lot less of that has been leached out of the oil over time. But interestingly, O-xylene, even though it has a very similar solubility to M-xylene, has dropped a lot in the oil, just as much as benzene. And so I'm going to explain why there's a difference. So on this, I just brought up circles of benzene, O-xylene, and M-xylene that were used for the illustration. On the bottom axis is effective solubility, and on this vertical axis is fraction remaining. And the dots are all of the volatile compounds that were analyzed in 2010. And so the first thing to notice is that more soluble compounds are more depleted in the oil, and less soluble compounds are still largely retained in the oil, but are still somewhat depleted compared to the starting composition. And then there's this interesting difference between O-xylene and M-xylene. They have the same solubility, but O-xylene is a lot more depleted. These are compounds that have been shown to biodegrade under methanogenic conditions. So those compounds have are more depleted because the bacteria living right next to the oil are munching on them, and as they deplete them, more dissolve from the oil. So there's this enhanced dissolution that occurs because of the biodegradation. And these compounds are also highly degradable. These are the N-alkanes of 6, 7 through 12 carbons, and they are quite depleted even though they're very insoluble. And we have concluded that the bacteria can degrade these without necessarily dissolving them into the water because the solubility doesn't seem to affect their losses. So the take-home message from this slide is that compounds are lost from the source at different rates that depend on the properties of the compound. So one might reasonably ask, well, I don't want to know about individual compounds. I just want to know how much of the fuel is still there. So it's difficult to say how much of the fuel is still there because there are hundreds of compounds in the case of crude oil. So you cannot measure hundreds of compounds. But there's a way to finesse the problem by measuring a single conservative compound that isn't lost. So this cartoon here is to illustrate how you would use a conservative compound. So at the time the oil spills, you have this conservative compound has a mass per mass concentration of 30%. So 30% of it has but then over time, this much, 30% of the mass is lost. And if you go back, but none of the conservative compound is lost. So then if you go back and measure the conservative compound, its concentration is now 50%. And so you can take the ratio of the original concentration, which was 1/3, and divide it by 50 by 1/2, and you're going to get overall 2/3. And so that's going to tell you that 2/3 of the mass is remaining compared to 100% that you started with. Or you can do 1 minus that and say 1/3 of it has biodegraded. So the compound we used is a triaromatic styrene. And by the way, each slide that has a reference, the reference is listed at the bottom. So for the Bemidji site, here are the results. Here are years since the spill. Here are the 13 samples collected in 2010, and we sampled them again in 2019 and 2020. And what you see is that about, so here's 40%, about 50% of the mass has been lost since the spill. This sample, we're not sure. I had this analyzed twice. I don't know what's going on with it. So I'll just say that if you ask me about it later. So the Red Hill spill was JP5, and what we'd like to know is, could we do this with JP5? Is there a conservative compound? And yes, so these highly branched alkanes listed here and are listed in this memo that's on the web can be used to estimate losses because they aren't lost very quickly. So these are recent results that are in this memo dated March 7th of samples collected from the sump and the Red Hill Shaft water gallery, the drain line. And so what you see is that they're not very different from each other. So that tells you two things. One is that they all represent the known spilled fuel. But there is one big difference right here. This sample from the water gallery has lost a lot compared to the others, and it's lost NC10, which it's lost to volatilization. So these conservative compounds can not only tell you how much mass is lost, but they can also tell you something about the processes that were responsible for the losses. So the second part of the talk is going to be about the contaminant plume. So this is just a cartoon that we published in a fact sheet, the source zone, the pipeline break, the plume is anaerobic, and then there's oxygen seems to slowly diffuse into the plume with distance down gradient. So I'm going to be talking a lot about dissolved organic carbon, and I'll explain to you why, how that compares to total petroleum hydrocarbon in a minute. So this is distance along the x-axis, concentration, and again, NDDOC is non-volatile to distinguish it from the BTEX and the petroleum volatiles. So coming in the clean water is about 1.4 milligrams per liter and about over 12,000 individual molecular formulas. So natural organic matter is incredibly complicated, and it really complicates our task of figuring out what is added in the source zone. So as the clean groundwater enters the source zone, concentrations increase to 30 milligrams per liter, and about 2,000 additional formulas are added. So the plume has over 14,000 molecular formulas. So if you just try to compare molecular formulas, you're going to see a lot of similarity between down gradient and up gradient. So we have to be more sophisticated in our techniques for saying, how is this organic carbon different from what came in? The good news is that this organic carbon that enters in the source zone does biodegrade quite rapidly in the aquifer. And so by about 200 meters down gradient, it's only about a milligram per liter over the background concentration. And then it pops back up here, probably because this well is maybe more in line with another, the main part of the plume. So it doesn't degrade completely. So there are some compounds left here that that sort of keep me up at night. And I'd like to know what is not finishing biodegrading. But one thing that's clear is that the added carbon reflects ancient carbon. So we know that it came from the oil because it's carbon-14 dead. So we'd like to know, what are these compounds and do they have biological effects? So I promised you I would relate this to total petroleum hydrocarbons. So here's non-volatile DOC versus distance. The orange is total petroleum hydrocarbons in the diesel range. The green is TPH in the gasoline range. And the gray is total petroleum hydrocarbons in the diesel range with silica gel cleanup. So Roger talked a lot about this last week. Silica gel cleanup isolates the true hydrocarbons. So what we're seeing is that the TPH is mostly these polar metabolites or biodegradation products. And here's the background DOC just for reference. So the plume is about 22% TPH in the gasoline range, but it's dominated by these NVDOC that's added in the source zone. So why are the analyses different? Well, non-volatile organic carbon is analyzed with a high temperature combustion method. So the samples are filtered in the field. That's why it's called dissolved organic carbon. Later, I'll be showing you total organic carbon, and the only difference is that the samples aren't filtered. Then the sample is acidified and purged with nitrogen to remove the inorganic carbon. And the volatile organics are removed. So that's why we know this is a non-volatile DOC. So then the remaining DOC is combusted and the outgas CO2 is measured. So that's why you want to remove the inorganic CO2 first. So that essentially gets you all of the DOC except the volatile DOC. To measure petroleum hydrocarbons, the first thing that's done, so the samples are preserved in the field, and then the organics are extracted with methylene chloride or hexane. There's a GC analysis, and all the chromatographic response between 10 carbons and 28 carbons are added up and compared to a diesel standard. So the things that TPHD doesn't measure are that partially oxidized compounds do not extract well with methylene chloride. And also some of the extracted compounds elute outside of this 10 carbon to 28 carbon window. But certainly the extraction problem is the most serious one. So this is a bar chart comparing 4 different extraction methods. Dichloromethane is what's used to do a total petroleum hydrocarbon analysis. And the total carbon that's extracted with dichloromethane in the Bemidji plume is shown here in black. The total carbon that's extracted with these other solid phase methods that are newer, and well, they're also, it's not just that they're newer, they're designed for extracting partially oxidized carbon because that's what works in the environment. So this method is what's used by people who study natural organic matter and its state in the environment. So the rest of the talk will concentrate on the difference of what you see with carbon extracted with this Oasis HLB solid phase extraction method, which gets pretty close to 100%, not 100, between 80 and 100% with 3 replicates. So coming back to Red Hill, we do have total organic carbon data for Red Hill. And these are data from monitoring well 2, collected between December of 21 and March of 22. And on the horizontal axis is total petroleum hydrocarbons in the diesel range. The vertical axis is total organic carbon. So what you can see is that, first of all, the most important thing is that the organic carbon is lower than for the crude oil site. So at our site, we had up to 30, and you have more like 10. So what that tells us is that these biodegradation products are present in lower concentration at this site. And I think that's good news because as Roger said last week, the dose makes the poison. And so to the extent that we need to know more about these partial transformation products, we have fewer of them at Red Hill. The TOC is not quite double the TPH, so there are things in the TOC that aren't captured by the TPH. There are a couple of approaches that that I'd like to pursue. One is to see if the total organic carbon drops with distance, and the other is to look at the carbon-14 signature of the TOC, because that way we can distinguish if it's TOC that came from the fuel or if it's natural. So what is this NVDOC? So partial transformation adds oxygen to the hydrocarbons in the oil, which creates polar bonds, which increases solubility. So here's naphthalene, so a polyaromatic hydrocarbon of 2 rings. It can react bacteria can catalyze a reaction with CO2 using this enzyme naphthalene carboxylase and form this naphthalene carboxylic acid. So that's just one example, and this is more soluble than the parent compound naphthalene. And in general, PAHs can be mobilized from the oil by these carboxylase reactions or other reactions that add oxygen functional groups. And we find oxy-PAHs in the Bemidji plume. I don't have time to cover it, but it is published. So overall at Bemidji, the overall average formula, so hydrocarbons would just have carbon and hydrogen, but we have these oxygens added. And the bond types, Roger mentioned this last week, that the GC work done by Don Zemo has classified compounds by bond types, and this was also done in 1998 for the Bemidji plume, where the compounds were classified by bond type, and this is percentage estimates of the different types. So we'd like to know more about what happens as this pool of carbon biodegrades, and we've applied 3 different advanced techniques. I'm only going to explain one of them in the interest of time, but the results of all 3 are published. So this one I'm going to talk about because I know that University of Hawaii has this capability, this EAMS capability, and I think it would be really great to brainstorm about what EAMS could be used for in the Red Hill case. So it's excitation emission matrix spectroscopy. And what happens is the sample is put in an instrument where there's an excitation spectra, a range of excitation spectra arrive, a single frequency that is applied to the sample, and then the emission of the sample is measured. And then the degree, the amplitude of the emission is plotted here as a color contour plot in this 2-dimensional space. And then each of the samples, so there's a 2-dimensional plot for each of the samples in the plume. And then the ParaFact, the parallel factor analysis, what that involves is that you take all the samples and you find a way to break down the data into a simple number of factors. And so each factor is a section of the chromatogram that can be used to recreate the spectra, or most of the spectra, for all the samples. So this is the 6 factors that were resulted from the parallel factor analysis at the Bemidji site. And now we're going to look at each plot is all the samples that were collected in the plume. So each black dot is a sample. And on the horizontal axis is travel time or distance from the spill. So travel time in years. So each plot is telling you the percent contribution of that part of the spectra to the sample, to each sample. So we start here with samples that are near the source, and there is a large contribution of C1, which is associated with reduced petroleum hydrocarbons. And that drops with distance, as does C5. So these two are associated with properties near the spill. And in each plot, the range of background values is shown in gray. And so as we move down, what we see is this part of the spectra, C3, there isn't much near the source. In C4, there isn't much contribution near the source, but as the pool of carbon migrates downward, we see an increase in C3 and C6. So these are more oxidized compounds compared to more reduced compounds near the oil. So the reason for using a technique like this is because, remember, we're talking about 14,000 individual compounds at least. So it's really astonishing that with just 2,000 compounds added in the source and then biodegrading with distance, we see this huge difference in the character of the pool of the DOC as it migrates down gradient. And that is telling us that the bacteria are effectively oxidizing this pool of DOC. So it looks less like it looked near the oil and more like natural organic matter as you get down gradient, but not completely like natural organic matter. So the last part of the talk is to cover the biological effects of the different fractions in the plume. And there were 6 wells that were part of this study. It was published in 2020. And I just compared the fractions that were extracted with the best organic carbon extraction, the fraction extracted with dichloromethane, so the TPH method, and then dichloromethane with silica gel cleanup. The method uses human liver cells. So if anybody read The Immortal Life of Henrietta Lacks, so these are, in her case, it was ovarian cells, but these are human liver cells that are used as a model of liver metabolism. So the way the method works is that the cells are transfected with dummy DNA that is activated and duplicated when a transcription factor is activated in response to an environmental contaminant. So this method tests for many transcription factors simultaneously, and the activities are reported as a ratio of induction values to control cells. So that's hard, that's a lot in one slide. Here's an example of how things are reported. So in this case, 52 transcription factors were tested at once, and only 2 of the transcription factors had big activation from this Bemidji sample very close to the source, with DOC at 16 milligrams per liter and TPH at 3.5 milligrams per liter. They were the pregnane X receptor, which is known to be activated by xenobiotics, and the aerohydrocarbon receptor, which is known to be activated in response to planar hydrocarbons, particularly PAHs. So the good news is, I guess, that this plume is activating those things that we expect it to activate. So now I said that I would compare the different fractions. So on the horizontal axis is DOC concentration, and on the vertical axis is how much the PXR transcription factor was upregulated compared to the control. So 1 means there's no activation compared to the solvent control. And here's the background well with not much activity. But then the green dots are the true hydrocarbon fraction isolated with silica gel cleanup. And we saw that that's very small at the site. So the true hydrocarbon fraction is not really responsible for what these liver cells are doing. This is DOC extracted with dichloromethane, so that would be the TPH fraction is in orange. And then this is DOC extracted with HLB. So what you see is that there is some activity that's not reflected in the TPH. In this case, not that much. But the silica gel cleanup is not really capturing that. If we need to worry about these, they're not captured in the silica gel cleanup fraction. So here's another way of doing the same test is this chemically activated luciferase expression. And in this case, we only tested the aryl hydrocarbon receptor, and we tested it again in human liver cells. So on this plot is the concentration of NVDOC for reference. So distance down gradient from the center of the oil, and the concentration of TPHD down gradient for your reference. And then on the right-hand axis and shown in red is the response of these cells that are human liver cells that glow at proportionally to how much the aryl hydrocarbon receptor is activated. And then this is normalized to beta-naphthaflavone, which is a known activator of the aryl hydrocarbon receptor. So you can see that the NVDOC is kind of aligns with the response of the cells, telling us that they're responding quantitatively to what is quantitatively present in the water. So this is a very complicated slide, but I just wanted to remind you that the concentrations at Red Hill are down. So somehow this slide has changed. So I'm going to back up. The concentrations at Red Hill are 10 to the 1, about 10. So Red Hill concentrations are down here, where these things are just starting to register. And concentrations drop with distance. So if we look at, sort of what's near the source, that's this orange. And values, what's interesting is that as concentrations drop with distance, the response of the cells drop with distance, as we saw here. So there's less biological activity with distance. Down here, the most down gradient well, it still has a big response, but that's only if you concentrate the carbon up. If you actually look at the response down at that well, at its in situ concentration, it's down here. So as these things biodegrade and their concentrations drop with distance, their biological response drops with distance. So in summary, the oil source, 86% of the carbon exits as CO2 surface efflux. And we see a lot of CO2 efflux at Red Hill. Losses differ by compound, so you can't really think of all of the oil going at once. It goes in fractions. And we see that at Red Hill too. Overall losses 50% in 40 years, and that's of oil. which has a lot more refractory material in it than jet fuel. In the groundwater contaminant plume, most of the non-volatile dissolved organic carbon is comprised of compounds not quantified by TPHD. And that appears to be true at Red Hill, although TPHD is a higher fraction of what's there. The good news is that these partial transformation products do biodegrade quite effectively, and they drop to 30 milligrams per liter. They do have biological effects, so there are more studies ongoing to determine if there are human health implications, because this is just a liver cell assay. It doesn't, the liver is processing this stuff, and it doesn't tell us if the whole organism would be affected. So that's all I have. I'm going to leave up my acknowledgement slide of all the people that have worked on the Bemidji site on the studies that went into this talk. And I'm happy to stay around for questions. I'll stop here. Thanks.
All right. Thank you so much for taking the time to join us in our seminar series. A lot of great information in that talk. So I think we'll open the floor to questions now. So earlier in the chat, I had 3 questions from Paul Ayer. So I'm going to go ahead and read those off one by one. Do you want to answer them one at a time, or do you want me to read them all at once?
So I haven't been reading the chat. What percentage is related to degrading petroleum hydrocarbons based on carbon-14 data? Yeah. What is the source of the remaining organic carbon? Are bacteria in the water included in the NVDOC data?
Dr. Bekins, sorry. There was a direct message question from Paul Ayer to me. So if I could read those to you first.
I misunderstood. Okay. Yeah.
Okay, so the first one is, could the contamination below the Red Hill fuel tanks be remediated by flushing the aquifer from above with a remedial solution through the perforated shallow angle borings that penetrate beneath each fuel tank?
Oh, that's a very interesting question. I have to say that I don't have good knowledge of the literature in practice. There is a very large industry flushing LNAPL and DNAPL from aquifers. The trick is to target the flushing solution adequately. So you need to make sure that it contacts the LNAPL. So the first step would be to find it. And then the other issue is that the flushed water has to be collected as it arrives at the water table. So that, and as we know from the GAC system, we are still in the process of evaluating where the water, how that collection system captures flow lines under Red Hill. Interesting comment.
Okay. The second question is, do you have a sketch of the degree and extent of contamination beneath the tanks drawn to scale with data from the history of monitor well data?
No, I don't. I wish I did. That would help.
One last question from Paul Ayer, and then can go into the public chat. So the last one is, why isn't TPH used to determine the mass loss rather than the trimethyls?
The TAS, the triaromatic styrene. So the reason why TPH isn't used is because TPH doesn't capture everything. You have to collect the TPH sample in the same location every time. So let's see, how to say this. So we have the saturation is so different. It varies from 10% to 50%. So if you just collect a soil sample, you're not gonna get the same TPH extracted from the soil as you would have gotten 10 years ago, 20 years ago, 40 years ago.
Okay, before I move on, actually, Paul Ayer followed up with, if you don't know about the shallow angle borings beneath the fuel tanks, he asked if Tom or Don Thomas or maybe Robert Whittier could maybe
I
jump in.
do know about the shallow angle borings. I was responding to the feasibility. I do know about the shallow angle borings.
Okay, yeah, that was his follow-up.
Yeah.
So move on to Roger Brewer's question. If you wanted to read them yourself, that's fine
yeah.
as well.
So I read Roger's, and everybody can read it for themselves. So the organic carbon that's added in the C14 data show that that all of the added organic carbon in the source zone is ancient organic carbon. The bacteria are not included in DOC because they are filtered out. Roger, you and I should talk offline about, I know there's a hypothesis advanced by Kirk O'Reilly that the DOC is biosynthesis products of the bacteria. And we have a new USGS report that looks at the difference in the DOC compared to biosynthesis products, particularly the nitrogen compounds, nitrogen-containing compounds. So Roger, you and I, this is of course a very complicated question, but basically everything that's added in the source zone is from the petroleum hydrocarbons.
Okay, the next
question is Mark. Was there any attempt made to stimulate or augment bacterial populations in the involved in biodegradation? If not, why not? So no attempt was made to stimulate the bacteria. The reason, so there are several reasons why. One is that the USGS management our instructions were to do basic research on the environmental processes, not engineering research. And that turned out to be a good bet by management because the natural processes have been adapted across the country. And it turns out that bacteria that can degrade petroleum hydrocarbons are ubiquitous in the environment, and even those that can degrade petroleum hydrocarbons anaerobically. And so the bioreactor was quite quick to, the natural bioreactor grew and was established within a few years. Having said that, there is an interesting effect where nutrients that are carried naturally from the surface, say, there's a lot of nitrogen deposition in Minnesota, ammonia deposition, because it's farm country, and that is carried rapidly into the subsurface at one end of in a topographically low area, and the oil has degraded a lot faster at that end because of this natural process of nitrogen deposition, atmospheric nitrogen deposition. Next question, Tao Yan. Given the geological, mineralogical differences between Red Hill and Bemidji, can we and how do we extrapolate the observed biodegradation rate from Bemidji to Red Hill? Well, let's see. There are several good things that Red Hill has going for it. One is you have a lot of sulfate. So that's going to make biodegradation somewhat better. you probably have a lot of iron. It's not named Red Hill for nothing. And so iron is an incredibly effective electron acceptor for biodegradation. The biggest problem with extrapolating rates is not the geochemistry, in my opinion. It's the groundwater flow velocity and the complexity of the aquifer, making it difficult to convince yourself that you were looking at the same package of water as it moves down gradient. So one idea, one thing that people have done to get around that problem at other sites is to create kind of a wall or a line of wells, so you capture a cross section of the plume, and then you sum up the concentration over a cross section. So instead of being in the business of saying this well flows to that well, you're in the business of saying this water from this plane flows to this water in this plane down gradient. Is a decrease of biological activity from the plume center a result of dilution of polars or due to different proportions of alcohols, ketones, et cetera, down gradient? Yeah, so it's not due to dilution. The paper that shows the three methods that show the difference in character of the DOC is one very important line of evidence that it's biodegrading. The whole pool of DOC is getting more oxidized. And the hydrogen to carbon ratio is decreasing. The oxygen to carbon ratio is increasing. The C14 data is also very compelling. I did not show it. Those data were collected as part of an agreement with Chevron, who's worked at the site. And Chevron has not yet published those data, but to me, they present a very compelling picture that what's actually happening is biodegradation rather than dilution. Al-Qadi says, I know this is outside the scope, but I'll ask anyway, what's the objective of the modeling effort? Oh yeah, a subject dear to my heart. So it was an overall mass balance effort. The work was funded by the Strategic Environmental Research and Development Program. And it was funded as part of a project to look at secondary impacts of bioremediation. And so the goal was to look at the fate of iron, reduced iron, which can be a co-contaminant, the fate of arsenic, the fate of CO2 and methane. And so the whole mass balance effort was to cover not just the loss of the hydrocarbons and the plume and the groundwater in the plume and the source zone, but also the fate of the biodegradation reactants that are part of the process, the methane, the iron being the main ones. But there's a couple of papers, well, you'll see, you saw the references, so I'm happy to talk to you about it more. Do I have thoughts about the most important research gaps to be addressed on the Red Hill issue? Yes. Oh, Paul, I don't know why, where I got that incorrect number. Thank you for that correction. I don't know where I got the incorrect number. I will check my slides.
Okay.
Do I have thoughts about
what the incorrect or the correction was? I think that might have been directly to you.
Yeah. Okay, so the most important research gaps. So I tried to cover those. I would like to know the nature of the total organic carbon. I mean, we have studied the heck out of crude oil, but we have no examples of heavy fuel. We have studied a gas station plume, and DOC is slightly larger than TPH, but not by much, and doesn't have biological effects. So that's really good news about the many gas station sites around the country. So we would like to have heavy fuel, an intermediate fuel like jet fuel, to look at these partial transformation products and their health effects. There are also many opportunities, I think, for developing new tools to deal with complex aquifers like this one, geophysical tools. I think those were, and then the CO2 efflux. I think that the CO2 efflux method has been used at the site, but it's very expensive. If there was a research component to come up with cheaper CO2 efflux methods, it could really turn into like a location tool for finding source zones. So I'd like to see some of that done. I think those are my main ones. I guess if there's no others, I would say that, the idea that there's a conservative compound, I think is really important. Everybody's struggling with how to assess whether the source zone of a spill has lost mass over time. And this idea of archiving the original spilled product and using a conservative compound, I think could be really a productive way in the future. So if we had a better idea of what compounds are conservative in JP5, that could be a research goal too.
Okay, great. If there are no other questions, then we can wrap up. All right, so just once again, want to thank Dr. Bekins so much for joining us. And thank you to the audience as well for taking the time to join us in our seminar. I'm glad to have you here.